Most adult-onset neurodegenerative diseases, and indeed childhood-onset diseases like lysosomal storage diseases, are associated with the formation of inclusions or aggregates within the cytoplasm of neurons. And there's extensive genetic and transgenic evidence that suggests that these types of aggregate-prone proteins, particularly in Mendelian forms of these diseases, exert their pathogenesis through toxic gain-of-function mechanisms.
The central feature of these disorders is abnormal protein aggregation, which leads to the development of insoluble cytoplasmic or intranuclear inclusions or extracellular deposits. The first of which is Alzheimer's disease, which, as I mentioned, is the single most common neurodegenerative disorder within this group of entities. And we will subtitle this portion of the talk, A Tale of Two Proteins. Of the two abnormal proteins, the first one that we'll discuss is tau. Tau is a microtubule-associated protein whose normal function is to stabilize the cytoskeleton of the axon. The other abnormal molecule that's present in the brains of patients with Alzheimer's disease is beta amyloid, or also called A-beta. A-beta is a 40 to 42 amino acid peptide which is cleaved from a much larger 695 to 770 amino acid precursor protein called amyloid precursor protein, or APP. APP is a transmembrane neuronal protein that may play a role in cell adhesion, although all of its normal functions are not fully understood. The key diagnostic features of Alzheimer's disease were described a little over 100 years ago by Professor Alois Alzheimer. He used silver stains, and these are silver stain images, much like he would have reviewed, demonstrating the two key diagnostic lesions, neurofibrillary tangles and senile plaques. On the left-hand panel are depicted some neurofibrillary tangles, which are the three flame-shaped structures that are present within the cytoplasm of neurons, often pushing the neuronal nucleus to the side. In the right-hand panel is a senile plaque, which consists of two components. One, a peripheral rim of dark staining cell processes called dystrophic neurites, processes of neurons that are affected by this disease. The central amorphous brownish substance is amyloid, which is an extracellular deposit of another protein that is found to be abnormal in this disorder. The second most common group of disorders in the neurodegenerative disease camp following Alzheimer's disease are the so-called Lewy body diseases. Lewy body diseases come in two major clinical subtypes, the first of which is so-called idiopathic or Lewy body Parkinson disease. The second subtype of Lewy body disease is so-called dementia with Lewy bodies. Lewy bodies were first described by Professor Lewy in 1912. Interestingly, he identified them not in the substantia nigra, where we recognize their clinical significance today, but rather in a basal forebrain nucleus called the nucleus basalis, a cholinergic nucleus that is heavily involved in not only Lewy body diseases but also in Alzheimer's disease. This picture indicates two Lewy bodies in a single neuromelanin-containing substantia nigra neuron of the midbrain. And in 1998, antibodies produced to alpha-synuclein were found to label Lewy bodies in cases of human Lewy body disease. Alpha-synuclein is, again, like tau, a normally occurring protein. It is a 140 amino acid protein that's localized to the presynaptic terminals of neurons. Its normal function is uncertain, but it is thought to potentially have a role in synaptic transmission. This is an immunostain section using an antibody to alpha-synuclein and a red chromogen in this case, staining a substantia nigra neuron that contains a dense aggregate of red staining material, which is a Lewy body. But it also shows that there is abnormally stained alpha-synuclein within the cytoplasm of that neuron diffusely, as well as in a number of neurites of that and other neurons within this portion of the substantia nigra. This is an example of the advantage of alpha-synuclein immunohistochemistry in identifying Lewy bodies and Lewy neurites in the neocortex of a patient who had dementia with Lewy bodies. On the left is an H&E stain section demonstrating a single neuron on the right-hand side containing a Lewy body in its cytoplasm. Cortical neurons, unlike substantia nigra neurons, do not have neuromelanin pigment in them, and this makes Lewy bodies much more difficult to identify in H&E stains of cortex. On the other hand, in the right-hand panel is an alpha-synuclein immunostain section of neocortex demonstrating not only large numbers of Lewy bodies, but also a dense feltwork of Lewy neurites in the background, all of these being pathological structures in this particular case. Next, let's discuss the frontotemporal degenerations, which I will subtitle A Tale of Many Proteins, because this is a heterogeneous group of disorders that may be characterized by the accumulation of lesions that consist of a variety of different kinds of abnormal proteins depending on the particular disease condition under consideration. Disease. In 1911, the first histopathologic description of PIC bodies was published, and this was actually an observation made by Alzheimer, who only a couple of years previous to that had illustrated the tangles and plaques that we now regard as the seminal diagnostic lesions of Alzheimer's disease. You can see that in the silver stain section of hippocampus that these dentate gyrus neurons contain ovoid dark staining structures about the same size as a nucleus. And again, these are the classic PIC bodies. In the late 1980s, it was discovered that PIC bodies, like neurofibrillary tangles of Alzheimer's disease, also contain significant amounts of phosphorylated tau protein, and this is an immunostain section demonstrating tau immunoreactivity within PIC bodies of the dentate gyrus of the hippocampus. Let's just summarize briefly the abnormal proteins that are present in the most common of the neurodegenerative disorders discussed so far. In Alzheimer's disease, we have abnormal tau protein, which is localized to neurofibrillary tangles, as well as to dystrophic neurites, not only throughout the neural pill of the gray matter, but also at the periphery of senile plaques. And we have beta amyloid, which is localized to the extracellular regions of senile plaques, as well as to blood vessels of the leptomeninges and cerebral parenchyma. In the Lewy body diseases, the primary protein abnormality is in alpha-synuclein, which takes the form of Lewy bodies within cell bodies, and it takes the form of Lewy neurites within processes of neurons. We discussed two subtypes of FTDs, or frontotemporal degenerations, that is the tau positive and the tau negative variety. Within the tau positive varieties, we had PIC disease, we had glial plaques and glial cytoplasmic inclusions and cortical basal degeneration, and we illustrated tufted astrocytes within PS.